Welcome to the Theoretical Neuroscience podcast. I am the host, Gaute Einvoll. One of the most important insights that has come out of computational neuroscience, at least I think so, is the idea of balanced neural networks. For recurrently connected neural networks, such as those found in cortex, a neuron receives approximately the same amount of synaptic excitation and synaptic inhibition from other neurons. What makes the neuron fire in action potential is typically a fluctuation away from this balanced, where excitation temporarily dominates. This is a bit unintuitive. When we drive a car, we push the gas pedal when we want to speed up the car and push the brake pedal when we want to slow down. We never push the gas pedal and the brake pedal at the same time. But this is what the brain apparently does when it comes to driving neurons. Here, the gas pedal, that is excitatory inputs, and the brake pedal, inhibitory inputs, are pushed at the same time. Today's guest, Nicolas Brunel, was one of the key developers of this balanced network idea in the second half of the 1990s. In Rome, with Daniel Amit, he wrote two seminal papers on the subject in 1997, complementing two equally seminal papers on the topic by Karl van Vreeswijk and Chaim Sompolinski in Jerusalem. In the 1997 papers, Nicolas and Daniel Amit investigated the dynamics of networks of excitatory and inhibitory spiking neurons, so-called integrated fire neurons, by a combination of numerical simulations and mean field theory, the latter a common tool used in statistical physics. In year 2000, Nicolas published a new paper where he took his investigations of the 1997 network, what is now called the Brunel network, a step further. Using a so-called population density approach, he was able to make phase diagrams of the different types of dynamic states the network would be in, depending on the values of model parameters such as excitatory and inhibitory synaptic weights. Network states in this context refer to whether the neurons in the network fire synchronously or not, and whether each neuron fires regularly or not. For example, in the asynchronous irregular state, what is referred to as the AI state, the firing of action potentials is irregular and also out of sync with the firing of other neurons. Such phase diagrams makes it much easier to get a grasp and an overview of the possible dynamics the network can exhibit. This 2000 paper was published in one of our specialized journals, Journal of Computational Neuroscience, and is one of my all-time favorite papers in our field. We have used the results in several of our own research projects, and so have many others. As of now, it has gotten more than 2,000 citations, which is a huge number for a computational neuroscience paper. In the podcast, we first talk about the history of how the balanced networks came about in the 1990s, with a particular focus on the 1997 and 2000 papers. We also talk about how the balanced idea is backed up by experiments and what advantages it brings to the functioning of the neural networks. We further talk about modern developments. After all, the idea is already a quarter century old, and of course also about what Nicolas has been up to since the 2000 paper. If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking, for example at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast, where you also can watch the videos. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners and hope that all of you can help spread the word about the podcast to other people in the community, on social media or otherwise. Okay, here is Nicolas. Yes, welcome to the Theoretical Neuroscience Podcast, Nicolas Brunel.
Well, thanks very much, Gaute, for having me.
Yeah. So first, before we start, this will be about the history and present status of balanced networks. But first, I thought I should we can ask you a little bit about where you work, where you are now.
Right. So I am currently in Milan, in Italy. I'm working in the Department of Computing Sciences of Bocconi University, which is a very recently created department. It's about, I think, exactly 3 years old now. And it's a very interdisciplinary department where you have people, mostly computer scientists, also lots of physicists working on machine learning, AI, and a small group of computational neuroscientists.
So you've been there for a couple of years, and then before that you were in US.
Right. So basically, I started. My first position was in Paris, but then I moved to University of Chicago, and then to Duke University, and then a couple
So
of years
you've
ago
been
back to
you're
Europe.
like a globetrotter in terms of, but your first position was in Paris, but
Yes, that's right.
before, I mean, you worked with, I mean, you took your PhD with Daniel Amit.
So actually, my PhD was officially from École Normale Supérieure in Paris, but
Mm-hmm.
I actually did part of it about a year and a half. I was in Rome at the end of my PhD. So that was the beginning of my collaboration with Dani Amit. And then
Mm-hmm.
I came back for a postdoc. So then I stayed 2 more years.
In
And of
Rome.
course, this was kind of a fundamental period for me. And it's kind of, yeah,
Yeah, that was like
great, great, great
the
epoch.
area of the invention of the balanced networks and the study of those. Yeah.
Exactly. Yeah.
So I think in balanced networks, I mean, when it comes to balanced neuronal networks is I think one of the key ideas when it comes to understanding of network behavior, at least cortical networks. So what is a balanced network?
Right. So, well, there are several ways of defining a balanced network, and we can go more into the details later. But the key idea is that you have a network where neurons receive both a very large amount of excitatory input, large excitatory synaptic currents, and also large inhibitory
Mm.
currents.
Yeah.
And the two approximately cancel each other. Because if they received only a lot of excitatory currents, then they will be all firing like crazy. If they
Mm.
receive only inhibitory currents, they will be silent. So the idea is that there is this idea of balance, the two balance approximately. And what happens then is that the network basically is mostly driven by fluctuations around
Mm.
the inputs. And so
Yeah.
I think that's kind of the key idea that,
Okay, yeah. Because it's a bit I mean it's a bit unintuitive in a sense. Because it's a little bit like driving with the gas pedal and the braking pedal at the same time. It sounds very wasteful,
yeah, yeah.
but that's how the brain does it in some sense, and it obviously has some advantages. Well, I mean, you can't argue. Yeah.
Yeah, well, I mean, I think there are still, it's still a key open question is basically why is the brain, in general, what is spontaneous activity
Mm.
that is observed
Yeah.
ubiquitously,
Mm.
in particular in cortex, but also in other brain structures.
Yeah.
What is it good for, right?
Mm.
And maybe these are
Yeah.
things that we can discuss later on
Absolutely.
because it
So,
looks like a little bit of a waste of energy that you have all these spikes that are emitted while maybe you're receiving no sensory inputs or
mm.
not performing any particular computation.
Yeah. So, because the key person here that you also mentioned was you sort of starting to work with Daniel Amit in the I guess middle late 1990s, but because he had already he had done some important work on attractor networks, right? He wrote this very nice book about attractor
Yes.
neural networks. And so, what was the state of understanding of cortical dynamics at the time when you started working on this?
Yeah. So actually, when I arrived in Rome, it was in '92, and it was a bit kind of a transition period because, as you said, so Daniel Amit had a uh, was a very important figure in the group of statistical physicists that got really entered the field after the Hopfield model came out, right?
Yeah.
And so a lot of physicists got really excited by neural network models. They realized they could apply all the kind of the toolbox of statistical physics of disordered systems that they had been studying. And so there was a kind of a burst of activity from 82 to, let's say, the end of the 80s, beginning of the 90s, mostly on networks of binary neurons. But most of the work was kind of a little bit disconnected from experimental neuroscience. Physicists working on these models, they were very far from people doing experiments in neuroscience.
Yeah.
And so at the early 90s, there is a small group of people, and Daniel Amit was like one of the first really that really started to try to bridge the gap between these models and the real brains, and started to interact with neuroscientists, and started to actually read experimental papers. I mean, it sounds a bit crazy now, but
Yeah.
when I started my PhD in Paris,
Yeah.
before I moved to Rome, I think I didn't read a single experimental paper before
Really?
moving to Rome, right?
Yes,
And so,
I
and
see.
so
Yeah.
I arrive in Rome, and then Dani Ahmed gives me, the book. I still remember the book by Breitenberg and Schütz, Anatomy
Okay.
of Cortex.
Yeah. Yeah.
They say you have to read this, and then
Yeah.
all kinds of experimental papers on persistent activity, including these landmarks, Miyashita papers. That was kind of at that point. This was the main thing going on
Yeah.
in Daniel's group. And so really there was this transition period where people were leaving these abstract models, these networks of binary neurons, which have been incredibly useful. I'm not saying that this was not useful. It's been extremely useful. But then kind of now trying to see now how can we now move towards neuroscience and really try to understand whether these models really do they tell us anything about what's happening in the
Because
brains.
one thing that when I read sort of your paper from that time, and it also that you were one of the first sort of to focus on this, just the background states, because one often, like the naive way of thinking about myself, right? You learn about the neuron, and then you need to depolarize it to get from the resting state to the
Yeah,
threshold, right? And then you think, well, if you just the way to approach networks is sort of to hook them up, and then you maybe try to drive it with some stimulus, but this thing of the focusing on the background states, or like the states where you don't really get any stimulus input, that was also
yeah.
something, I guess, a key realization that we need to understand the background states before we can understand the stimulus evoked states.
Yeah, exactly. So I think until the beginning of the 90s, no, at least, I mean, people from that community, the statistical physics community, I don't think that anybody has really thought of this as a major
No.
problem. And also part of it was because people were not really looking at the data. And
Mm.
so when you start to look at recordings from cortex, it's obvious that you have this incredibly ubiquitous spontaneous activity everywhere. And then you ask the question that, yeah, why is the network in that particular state in the absence of any input? So it looks like a ground state of the system. And
Yeah.
so Dani Amit was basically, I think, one of the first to ask the question. Well, the first question is what are the mechanisms, right? How can a network be? It looks like a very stable state,
Yeah.
and how can networks in cortex be in that kind of state where neurons fire irregularly at low rates, correlations between neurons seem to be weak, and so on and so forth. And so basically that was the main issue I started to work on when I came back for my postdoc in '93. So that's basically,
Okay.
and Amit had already thought about it a little bit, and so
Mm.
we,
Yeah.
so we
And this postdoc resulted in this quite sort of, I would say, very like seminal paper, and this in my head in '97. I guess you had two papers, one in
right.
cerebral cortex and one in neural networks. But
Network. It's the
neural
journal is
network.
Network Computation Neural Systems. It was actually the journal that was founded by Daniel Amit.
Oh, I see.
Yeah.
Neural network. Okay. Anyway, but I sort of particularly looked at the cerebral cortex ones, and so there you introduce this key idea of balanced excitation in and in spiking networks. So why was it important, do you think, to Well, I mean, dealing with spiking networks is mathematically a hassle, right, compared to dealing with continuous models. But you were able to deal with it in spiking networks, and so why was it important, you think, to have spiking networks?
Right. So yeah, so that was also the big part of this transition in the early 90s, where like moving the focus from networks of binary neurons towards networks of spiking neurons. And so basically we're interested in trying to reproduce the statistics of firing of cortical neurons. So it seemed like we had no other choice but to have spiking neurons as the basic uh, units of the network. And actually there were a number of people that started, I think, around well, of course, there were a few pioneers even before that, but at the beginning of the 90s, some people from different groups started to simulate networks of spiking neurons, mostly leaky integrated fire neurons. I mean, apart from, I mean, they were like a separate community working on more biophysical models. For example, people studying thalamocortical networks, like I'm thinking like Alan Destex, Terry Sejnowski, John Winslow, etc. But really, for these kind of cortical network models, there were a number of people that started doing simulations with these models, networks of leaky integrated fire neurons. But then we thought that we would try to go beyond just doing simulations, but trying to do some calculations. I mean, given that we were coming from the statistical physics community that were using mean field for all kinds of problems, and so that's where that was the challenge that we tried to address. And so so the idea was that so the first question was, how do you describe analytically these kind of networks? And so and I don't know if you want to to go into
No, I mean it's
now
because it's, I mean,
or later.
because one thing, no, this is fine, because this is, I mean, of course, the one approach is to put a network of integrated fire neurons on a computer, and then one thing that you realize then, in order to get something that looked a bit like this, I guess, what is now known as the AI state, the irregular and asynchronous spiking activity state, then you need to have this balance, so that on average you get them as much input from the excitatory neurons as on inhibitory neurons, and since inhibitory neurons are fewer, you need like a larger weight. And then, so that was obviously a cool thing, that you could sort of see that you sort of got sort of like a pattern, but you also use this, as I said, mean field theory. So you went beyond just simulations, right? So you developed this mean field theory, which was sort of like an idea from statistical physics. So what is a mean field theory?
Right. So it's actually, strictly speaking, it's not mean field theory, because you have to go beyond the mean, right? You have to
Okay.
capture the fluctuations beyond the mean. So basically, you have to extend mean field to also describe fluctuations around the mean.
Like the
But so
variance.
the basic idea was the following. So essentially, so the idea was to try to calculate self-consistently the firing rates of different populations of neurons in a network, with characteristics that were roughly consistent with experiments. So the idea is we took a randomly connected network with low connection probabilities, and then excitatory and inhibitory neurons. And then basically we tried to, we were particularly focusing on parameter regimes that were similar to cortex. So one important parameter is the number of inputs per neuron, and these are typically about 10,000 or several thousands in cortex, so very large. And then the amplitude of postsynaptic potentials are pretty low in average compared to threshold. So these were the kind of numbers that we had in mind. And then what
And
you realize
this is something maybe you got from the literature, like Breitenberg and Schütz, and
exactly. So the value, the number of connections from Breitenberg and Schütz, basically, so the basic numbers of the anatomy, and then for the strength of the connections at that time was when people were just starting to do paired recordings in slices. So there were like early work by people like Alex Thompson and others. They were starting to measure amplitudes of single excitatory and inhibitory PSP, postsynaptic potentials, and so we knew they were in average of order maybe fractions of millivolts, something like that. So that was also an important
So, because one thing I remember when I got into, well, learn about the computational neuroscience, that like the first papers I was sort of given to by my friends were sort of like this, like the Wilson Cowan paper, I guess from '72, and also this Amari paper, but they were, as I or maybe this is they assume this like mean driven inputs. Is that correct to say?
Yeah,
Not
yeah. There are no really fluctuations. So these are
no.
kind of the standard
Uh,
rate
yeah,
uh,
yeah, exactly.
rate model approach. So there are the different names for this family of models, but the idea is that you're describing the
Mm,
whole population of neurons by a
yeah,
firing rate variable, and
yeah.
then there
So.
is not really a description of the fluctuations
Mm.
around the mean
Yeah.
in these
So
models.
this, because this was sort of like a key insight then, that you sort of you need also to include the variance, and in
Exactly.
fact like the
Exactly.
most interesting regime is actually the variance driven regime, not the mean driven regime.
Exactly.
Yeah.
Exactly.
Yeah. That was
That's kind of the key feature.
yeah.
And
Mm,
yeah, so basically, the idea is that when you look at these numbers, and if you assume, so we had to make a number of assumptions to
mm,
make progress analytically, because in general these networks are
sure.
very hard. It's very hard to do exact calculations. And so the key simplifications is assuming that neurons fire as Poisson processes. So you
Mm,
start with this assumption, and then you have to check then that the
mm.
state that you get
Because
is
this
actually
was an experimental observation also of these cortical neurons, that they are like quite Poissonian.
right. Exactly. So
Mm.
they fire very irregularly close to Poisson processes. It's of course never exactly a Poisson
No, no.
process, I mean, for obvious reasons, but it's
Yeah.
close to. Then the other assumption is that we assume they are approximately at least independent. And this is, we justify this by sparse connectivity. So if the connectivity connection probability is small enough, then we reason that we could have approximately think about the neurons as being uncorrelated. Now the idea is that then each neuron, um, then you're using the two other assumptions that the number of inputs is large and the size of the postsynaptic potential is small. Then you can approximate the synaptic inputs to neurons by a Gaussian process, right? It's
Mm.
called diffusion approximation.
Yeah.
So
Mm.
it's well known. And then each neuron, the voltage of each neuron is technically speaking is basically becomes an Ornstein-Uhlenbeck process
Mm.
whose moments depend on the firing rates of the neurons of the neurons in the network,
Yeah.
which are still to be determined at this point.
Mm.
Then what you have to do is to then calculate what's the firing rate of a neuron, which is essentially an Ornstein-Uhlenbeck process. So the problem of calculating the firing rate is basically a problem of it's a so-called mean first passage time problem,
Mm.
the time it takes to cross a threshold, starting from the reset.
Mm. Yeah.
Then you calculate the firing rate, and then basically you get self-consistent equations
Mm.
where for the firing rates of
Yeah.
in our case were just two populations, excitatory
Mm.
and inhibitory.
Because you can get an analytical formula, I think, from for the firing rate, given that the input is has a particular mean and a particular variance, right? And you get this some kind of formula,
Right.
right?
So
With some.
exactly. So you have an exact expression for
Mm.
this, which is actually pretty old. It's known
Yeah.
from at least the 50s, 1950s. It's basically
Mm.
this first passage time
Mm.
calculations for
Yeah.
Ornstein-Uhlenbeck processes,
Mm.
which
Yeah.
basically were already known a
Mm.
long time before. We just used them in that particular context.
Oh. So I mean, this is like a little bit. I just read this. There was a paper out just about like celebrating 50 years of spin glasses. I don't know if you saw that. So this is sort of, I guess, the first spin glass models came in well, 75, maybe then, if it's 50 years. So
Maybe the Sherrington-Kirkpatrick model, probably.
yeah,
Yeah.
that was, I guess, one of. I think they wrote this this review or this perspective
Mhm.
paper also.
Mhm.
So so this was sort of like this exciting idea in statistical physics did, and it's like this thing that you have, I guess, is spin glasses. Then you have this disordered organization of spins. Some are up, some are down, and they are sort of frozen. So that's why I call it spin glass. So did they sort of did this kind of this kind of this did model have some inspiration for you, as you can remember, or
Well, I was actually quite familiar with all this literature coming from, because the group where I did my PhD were lots of people were working on spin glass models.
Okay, I see.
But the methods that we used, so there are a number of key differences, because so spin glass models basically they have the interactions are symmetric, right? So you have
Yeah.
there is a symmetric interaction matrix, and in the kind of networks that we are studying, of course, it's like not symmetric at all. You have excitatory inhibitory neurons, and even between
Yeah.
excitatory neurons, we took a random Erdős-Rényi directed graph, so it was not it was completely
Yeah.
asymmetric. And so that then you can't use really the same type of methods that you can use. You can't write down, Hamiltonian and then apply the standard two depths
The
of the.
things we like to do.
So you have to invent something new, basically. But that said, it's, I mean, I think it's still I think the kind of dynamics that people see in spin glasses, where you have these very slow dynamics in the low temperature region, it's actually still an open question whether this kind of phenomena could potentially be observed in networks of neurons. There are actually a few recent papers of people arguing that maybe if you, of course, cortex or any neural system is you never have symmetric interactions, but if you have some partial degree of symmetry, which is known to be true in cortex at least,
Yeah.
then you could have maybe this kind of phenomena could actually show up. There are a few very recent papers on this issue. So
Yeah. There
yeah,
was also this. Yeah.
yeah, no,
There was.
sorry.
Yeah. No. No, I don't. Yeah, that was more like a side comment. But there was also this like quite parallel work. At least some of these ideas was the work from Van Vreswijk and Sompolinski, right, with this chaotic balance state, and that was
Yes.
about the same time, right?
Yes, very much.
Yeah.
Yeah, we're very much competitors on
Oh, really? Yeah.
well, actually, I personally had no idea that anybody else was working on these kind of questions
No.
at the beginning of the project. I really thought that we were pretty much the only ones working on this.
Yeah.
But of course, there were some. So then we there were a few papers that came out that show that at approximately the same time, multiple groups were starting to think about this problem of irregularity in cortex. So there is a very famous paper by Softki and Koch where they pointed out that this irregularity was completely inconsistent with a neuron summing just a lot of excitatory inputs, because then
Yeah. Yeah.
you would never see any irregularity. And then there was a paper by Shadden and Newsom who pointed out, well, but you also have inhibition.
Yeah.
But of course, these models did not really explain how a network could stabilize in this kind of state. They were
No.
just,
Like make
pointing
it
out the problem
self-consistent.
and then a potential
Yeah.
solution. Then there was a paper by, Misha Tsodix had been a postdoc of Danny Amit just before I arrived, and he sent a paper he wrote with Terry Sejnowski about, I think the title of the paper is something like fast switching in balanced network models.
Okay.
So where basically they had simulated a network of excitatory and inhibitory neurons, and basically they found that if you had balance, that the network has all kinds of interesting properties, including this fast switching, which is one potential benefit of balanced networks, that they can react very fast to any input. And so we realized that, okay, there were lots of people thinking about this. And then for us, it took a lot of time to converge to basically also paper first manuscript getting rejected from Journal of Neurophysiology, which is kind of
Really?
a funny
Yeah.
story.
Yeah.
20 years later, at the time it was not very funny, but
No. No.
and
Yeah.
then at some point
Yeah.
we received this. We saw that Carl von Frieswijk and Chaim Sokolinski had just published a paper on science, and it's like,
Yeah.
and we thought, okay,
Yeah.
looks like
I
we were
see.
scooped.
Yeah.
But I mean, their model was, of course, I mean, this is a beautiful paper. I think it's one of the most beautiful papers in the field, and they studied a different model, which were a bit simpler, And they also had this key idea, this scaling of the weights with the number of connections, that you can do the large K is the number of connections. And then you can do a lot of calculations where you don't really need to do any approximations as the one we had done. But that model was definitely simpler because it was binary neurons where technically, when the input to neuron was crossing a threshold was like that would be called a spike. But it's simpler than integrate and fire, which allowed them to do.
But Amit he was associated with the Hebrew University where they were, right? Or
yeah. So at the time, he was spending 6 months per year in Jerusalem, and then 6 months in Rome.
So you think that they had some kind of, it's a possibility to exchange ideas or to
yeah. So my understanding, I was in Rome, so I don't know what happened in Jerusalem. Was that basically they got the idea from basically Daniel maybe giving a talk, and then they thought, there is this, we can study this in a simpler model, and then basically they published it. But I think that's my understanding of what happened. I have to say this is an amazing paper, and there is a companion paper in neural computation where they gave all the details.
A couple of years later, I think. Yeah. But one thing
there was all this activity at this time about balance networks. Of course, it has continued later.
Yeah, but this was sort of like a frenzied upstart. Yeah. But also, one thing you did was also probably reflecting that the background Amit in like associative memories. That you also immediately looked at how, if you are in this AI state or this a chaotic balance state that Van Resvik and Sompolinsky, I guess, call it, we can call it AI state. You not only showed why it's this background state is sort of stable and you can sort of a network finds this state itself. You also try to store some memories in it, right?
Yeah, that's right. So because this was really the continuation of previous work by Amit and collaborators, where he was very much, he was really laser focused on this issue of persistent activity as a mechanism for working memory, and also trying to really understand whether the attractor dynamics, which is basically what happens in Hoffield models and generalizations, is compatible with observations that in these experiments in primates, that there were like lots of papers coming out at the time in primates documenting personal activity. And so basically we wanted to check whether a network model that could generate this background activity, which is seen in cortex, can you structure synaptic connectivity in a way to generate these attractor states, or the memory states also. You can think about it as excited states, analogy with physical systems, where you have a ground state and then you have these excited states. So can you have coexistence between the two, as seems to be the case in experiments, where basically in experiments, the monkey has to do a delayed response tasks. So he's starting before the cue is presented. You see, background activity, and then the cue is presented, and then some neurons get persistently active, small fraction. Most neurons are not very much activated. And so the idea, can we see the same phenomenology in these networks? And the answer was yes, we showed that.
But then you added some synaptic plasticity, some
right. So the way we structured connectivity, so we had a very simple setup where there were like subgroups of neurons that were selected for specific stimuli. Let's say you have stimulus A, that strengthen the excitatory connectivity within its subgroup, kind of a Hebbian mechanism,
So you had
wire together, and then you have to balance things by decreasing connectivity between groups in order not to cause instabilities, and that was enough to generate persistent activity consistent with observations.
So I guess that was one of the second paper in neural network that was focusing on that was focused more on this and this memory thing, or
Well, I mean, both papers, you can find both spontaneous and personal activity in both papers. The
okay,
cerebral cortex paper, we really just showed calculations.
okay,
And then the network paper was focusing more on simulations, first showing that indeed, because the calculations, you make a lot of approximations. So
mm, yeah,
and also we didn't have a good way of checking the stability of these
mm,
states, and that
mm,
came later. I mean, the
mm,
later papers, which we might uh,
mm,
might talk about later.
yeah, yeah, yeah,
But so the idea is we wanted also to do simulations to see whether that actually works as predicted by the theory. And
mm,
so, and of course, in the simulations, you can also check that, for example, are
mm,
neurons really Poissonian or Poisson-like, and also we could look at cross correlations between neurons, which was completely out of reach of the calculations, at least at that time.
mm,
And we also, in that paper, we've also found this interesting observation. We also extended calculations in that paper to show that in these states, the distributions of firing rates across neurons is generically very broad. So when you have in a balanced state, you kind of automatically get very heterogeneous firing rates.
mm,
And this is
yeah,
something that's been observed
mm,
again over and over again in cortex, that
mm,
all the published distribution of firing rates in cortex have very broad,
mm,
typically can be well described by log normal
yeah,
distributions. And that's something that comes out automatically of this model. You don't really need to
mm.
do any tune any parameters. So.
So and then yeah, we had this 1999 paper with Vincent Hakim, and we're coming a little bit back to that, but I'd like to jump to I think my favorite paper of yours, and it's not only my favorite paper. I sort of like it's more than 2,000 citations, so that's quite unusual in I think in computational neuroscience. So, but because this was this paper in a journal of computational neuroscience, where I mean you still studied the same, more or less the same model, but you the integrate and fire model, integrate like these populations, and then, but you added this very important thing, I think, which are very well suited for human brains when you want to understand things, is like these phase diagrams.
Mhm.
So then you added this new technique, I guess, where you use this population density approach, this Fokker-Planck equation, which is sort of like another way to describe neural dynamics in populations, which are then borrowed from statistical physics. So this you had already used in this paper, I think, with Vincent Hakim in 1999, where he looked at oscillations. So what is this population density approach, and why was it so useful in this 2000 paper?
Hmm. Right. So, I mean, the motivation for doing this was that. So when I so, so first the calculations I had done in Rome with Amit were basically just looking at stationary states where we're assuming that the firing rates of neurons are basically constant in time, and
Mm,
that was kind of the only situation that we could study analytically. But then in simulations, I realized that if you simulate networks of excitatory and inhibitory spiking neurons, more often than not, you find that your network starts oscillating like crazy, right? You get all
mm,
kinds of synchronization phenomena. It can be either very strongly synchronized, or you can have also these more subtle, more weakly synchronized oscillations that can happen, especially in randomly connected networks.
yeah.
And so I was really, at some point, I kind of got really obsessed about, trying to understand what was going on. And the question is like, how can we characterize this analytically? And so
Mm,
that's where this idea of population density approaches um,
mm,
arrived. And so basically what I realized is that you could describe again, assuming that different neurons are, you can think about them as uncorrelated um, uh, the voltage of each neuron is basically an uncorrelated Ornstein-Uhlenbeck. But now potentially you can have
Maybe you can
situations
just
where
say what is Ornstein-Uhlenbeck for the
yeah,
listener.
so Ornstein-Uhlenbeck process is basically a particular stochastic process which where it's basically, you can think about it as a filtered white noise or a filtered Brownian motion. So this is the kind of the simplest stochastic process where the variable has kind of a non-trivial correlation time, which in single neurons would be the memory time constant. And so
Okay.
um,
So it's
and
a noise process that sort of is sort of suitable for the purpose.
yeah, exactly. So the idea is that you have the synaptic inputs, you describe them as basically a white noise process. So basically, it's a delta correlated, so there are no temporal correlations. But then, because the voltage is described by the standard voltage equations
Low pass
with
kind
the
of thing.
capacitance and the leak terms, you get low pass filtering of the white noise,
Yeah.
and that's Ornstein.
Yeah. Okay, good. So now we have Ornstein-Uhlenbeck in place. So now we can continue.
Right. So the idea is that to describe these states, now clearly in these states, the firing rate of neurons is not constant. I mean, the instantaneous firing rate, where the instantaneous probability that a neuron is firing, if you have an oscillatory state, then this probability is basically oscillatory. And so now you have to, so now the idea is that you have the Ornstein-Uhlenbeck, but with time-dependent moments, mean and variance. And then you can still describe, so the idea is now, if you consider the distribution of voltages of the neurons in the network, this in practice becomes described by a Fokker-Planck equation with time-dependent so-called drift and diffusion terms, right? So the drift is like how the mean inputs drive the membrane potential. It's described, and so this would be basically an oscillatory. If you have like an oscillatory input, then the voltage will also be driven in an oscillatory fashion. But then you have the diffusion term, which also describes how the fluctuations are affected by this oscillatory term. So basically, it's kind of this complicated nonlinear partial differential equation. And then, but of course, there is there is one simplification that arises when the firing rates are constant in time. So these are the asynchronous states, right? The idea is that then neurons are really uncorrelated because they receive this. Basically, each neuron has an uncorrelated fluctuation term. This we can write down the solution of this in this asynchronous state, and we can write down the firing rates, which basically the same firing rates as in the cerebral cortex paper. But now the nice thing is that, and that's what we realized when I started to collaborate with Vincent and Kim, I was back in Paris at that point, is that we can also do some stability analysis of these states, right?
Yeah.
And amazingly, we can actually, you can do this analysis analytically exactly. I mean, write down equations for the stability of this state. And what you realize is that, especially in networks that have strong inhibition, you have these instabilities that arise because of inhibition that are typically oscillatory instabilities. So they're basically Hopf bifurcations. And you can find out where these bifurcations are in parameter space and compute the characteristics of these bifurcations, like what's the frequency of oscillation. You can also do, so in the 99 papers, we did all kinds of calculations. We can derive the normal form of this bifurcation. We can.
So what's the normal form?
So the normal form was typically when you have a bifurcation of a particular type, let's say a Hopf bifurcation, then you can reduce the bifurcation to its essence in a way. So what's the simplest possible equation that describes this bifurcation? And so the idea is that you're really describing well what happens with a much simpler equation. This much simpler equation describes well what happens very close to the bifurcation point.
Exactly. So the thing is that in 1999 paper, you did this analysis on a simpler model because you only had inhibition. So that
Yeah,
was like
so we thought so we were yeah so that
Mm.
we thought that it would be a good first step. The calculations were already quite complicated for
Yeah,
just
yeah,
one calculation, so we thought let's start with the because we
yeah.
realized that this basic phenomenon that you could have asynchronous states, asynchronous irregular states, so
Mm.
you could get them in the network with just inhibitory neurons
Yeah.
driven by excitatory inputs. So that's the
Mm.
simplest possible balanced network. In fact, you
Yeah.
have just only inhibitory neurons, they're driven by strong external inputs, and
Yeah.
they inhibit themselves, and you get
That's
balance.
it.
Now
Yeah.
it's balance between the purely
So they only
external.
get excitation from the outside in some
Exactly,
sense. Yeah.
exactly,
So it's not like they're not really doing it. No excitatory neurons as such in the model. Mm. Yeah.
exactly.
So then you combine then you were bold and combined the model from 1997 with the methods from 1999,
Exactly.
and you did this work in the 2000 paper then. Yeah.
That's right. Yeah,
Mm.
and yeah, and then I mean you get
More.
richer behavior compared to the purely inhibitory network. I mean, you get more
Mm.
instabilities. Of
Because
course,
yeah.
you can get states, you can get instabilities to states where
Mm.
neurons fire like close to their maximal firing rate. So you can call
Yeah.
them epileptic states, if you
Yeah.
like. Kind of,
Because you had, I think you divided them up into four different types,
yeah,
where this AI
yeah,
was one of them, and then it was like asynchronous or versus synchronous, and that has to do with how the different neurons are, how correlated the neurons are to each other, and then you have irregular and irregular, which then
yeah,
tells you how well or how regular the firing of each neuron is, right? So you get
right.
these four types.
Yeah, exactly.
Yeah.
Yeah, yeah.
And then you get with all, yeah. So then synchronous regular, then that's essentially a very oscillatory, right? That's like a so all this.
Synchronous regular, it's kind of this what when people think about synchronized states in networks of neurons, that typically what the first state that come to mind is that all neurons kind of fire together in a
Okay.
periodic way. It's kind of
Yeah.
this boring state where all neurons are firing.
Okay.
Um, or they can break sometimes in cluster states. So that's another type of state where they break maybe in two clusters and they fire in alternation.
Yeah.
But these
Because
are
yeah.
yeah, in those states, basically neurons are firing very regularly, right?
Yeah.
Indeed.
So that's maybe like a epileptic seizure, maybe, or I don't know, or we can think of it's like you get strong oscillations, right? At least
Yeah, and the very high
frequency. Yeah.
activity, and also the network will be
Yeah.
totally sensitive to inputs in
So
this state.
and then you have synchronous irregular. So
Right.
what is
So
that?
that yeah, exactly, and that was actually what we were really focusing on with already in that paper with Vincent Hakim. We
Okay.
wanted to understand because these are kind of oscillations that are a little bit weird because typically people who study couple oscillators in many different systems, they study oscillations where the individual units, they oscillate at some frequency and the network synchronizes at that frequency, right? You have just one frequency for the. But here, and that's something that I had found in simulations,
Mm.
is the two are completely decoupled because the network can oscillate, let's say, at 200 hertz, but the neurons can fire at 5 hertz, right?
Ah.
And the idea, and so that's kind of a more weakly synchronized state, and so also that
Ah.
leads to the name synchronous irregular, because what happens is that neurons, their mean inputs are oscillatory, but they have large fluctuations around mean. So at each cycle of the oscillation, there is only some probability that they make it to threshold, right?
Mm.
And so that leads to the lower firing rate. So typically in those states, single neuron firing rate is lower than the population oscillation.
Yeah. So is that because this AI asynchronous irregular AI state that is we think sort of like seen in cortex quite commonly, or maybe more like the default state. But when you have oscillations, is that like this synchronous irregular state? Is that typically when you see something oscillating in the brain?
So I do think so, and the reason is that so if you I mean at least in many contexts. For instance, let's consider as an example 200 hertz oscillations that you see during sharp wave ripples in
Mm.
in the hippocampus. So these are really fast oscillations, right? 200 hertz.
Mm.
But single neurons certainly don't fire at 200 hertz during these
Mm.
oscillations. If you look at the neuron recordings, I mean, pyramidal cells, they might
Mm.
go up to
Mm.
five spikes per second. I mean, they do increase their firing rates. And then inhibitory interneurons, they also fire maybe 30 spikes per second in average, but at
Mm.
most. So, in fact, this looks perfectly consistent with these synchronous irregular
Mm.
states.
Yeah.
Likewise, gamma oscillations in cortex. I mean,
Mm.
most neurons don't fire at 40 hertz, right? There might be a few neurons that do this, but if you look at the population
Mm.
activity,
So
and so
that's
yeah,
also.
I do think that actually these states are actually a good description of what happens in at least some oscillatory states in vivo, and also
Mm.
the fact that it's another observation is that in these simple models, you typically move from asynchronous to synchronous state. I mean, from AI to SI by increasing external inputs, and that's
Yeah.
also consistent with in the sharpwave ripples. It's known that there is this big increase of inputs to area CA1 from area CA3, and the firing rates of most neurons increase.
Mm.
And
Mm.
also in sensory areas, when you see gamma oscillations, that's
Mm.
typically when you have sensory inputs present. So
Mm.
I think that's perfectly consistent with
Yeah. And what's
the.
great about this paper is that you actually show a bunch of these phase diagrams, meaning that what states are you in for
Yeah.
different values of these key parameters, like the external drive and like I guess the balance factor, how balancing.
Right.
So this is whenever you get the phase diagram, then it's much easier for humans to to get a grasp on it. If not, it's just like flying around in parameter space without the phase diagram in the end. Then at least my brain is not well suited to deal with that. It's quite so that was really cool. So, and one thing you sort of also show that this, if you start having this some spread in the delays, synaptic delays between the neurons,
Yeah.
then some of these like more delicate structures vanish, right? So it's a way to, at least this synchronous regular seems to be, then you only end up with these asynchronous regular states. Or
Right. So in general, introducing distributions or heterogeneities of delays tends to stabilize the asynchronous state. And it
yeah.
was in some cases, in some of the bifurcations that I showed in that paper, it has really a uh, very pronounced effect. You're
Mm.
right. The general effect for introducing heterogeneities in delays is that it stabilizes
Yeah.
asynchronous states. So it becomes harder for the network to oscillate when you
Mm.
have broad
Yeah.
distribution of delays. Yeah.
Mm. Cool. So the key trick here was sort of to get these phase diagrams. Was sort of that you were able to look, well, formulate into this Fokker-Planck thing, the population density, and look at sort of when you had these bifurcations, which means that you change some model parameter and suddenly the behavior becomes qualitatively different, and you go, say, from like the AI state to the SI state. So that was sort of like the what made it possible for you to make these phase diagrams, right?
Mhm.
Hmm. So yeah. So then, I mean, what are then the, what do you say, the evidence for this excitation inhibition balance
Hmm.
in experiments? How do you convince the
Yes.
experimentalists about this, or yourself?
Right.
Yeah.
Okay. So I would say, I mean, I think what's remarkable with this model or this, let's say, family of models is that it, in a very parsimonious way, it can explain lots of different observations, right? I mean, the irregularity of firing is basically you get it automatically. Broad distribution of firing rates also. There is evidence. There is some evidence of balance between excitatory and inhibitory inputs from intracellular recordings
Mm.
in cortex. I think it's not a lot of evidence, but there is
Yeah.
definitely some people have claimed that these recordings are consistent with balance between excitation and
Yeah.
inhibition.
Because this must be in vivo then.
Oh yeah, yeah, yeah. These are in
Yeah,
vivo. Yes,
I mean,
yeah.
it's difficult to test in vitro, I guess. At least this aspect is difficult to test in vivo or in vitro. But I guess you need to be in a in vivo network and in living animals, and then see that this
Yeah,
is
so
actually
there
holding.
was actually this really nice experiment. That you're mentioning in vivo versus in vitro, there was this very nice experiment by Alex Reyes and Barral, who's the first author, where they did this. They looked in cultures and they could basically activate this culture. I don't remember how, but basically they found that a lot of the characteristics of balanced states they could reproduce these in cultures. There was another kind of really nice observation is that so in balanced networks, when you really take this limit when the number of connections k goes to infinity, then you realize that for the fluctuations around the mean to be of order one, so not to be neither really large or really small, then the amplitude of the postsynaptic potential should scale as 1 over square root of k, where k is the number of
Mm,
connections. So that's a really
mm,
simple calculation to derive this relationship. And what they did is that they used cultures with different densities of neurons, so they
mm,
could actually vary k, and then they measured
oh,
the strength of the EPSPs, uh, and they showed that actually there was something like a 1 over square root of k relationship,
mm,
as if these networks were trying to self-organize, to maintain this balanced state. And
mm,
I think this is the only example I know that of an in
yeah,
vitro preparation testing some of
mm,
um, the
mm,
predictions of the balance network. But I thought it was a very very nice paper.
yeah,
There were also papers from David McCormick and collaborators where they took this preparation where you can induce up and down states. And I think what they showed is that in the up states you have this balance between excitation and inhibition. So that's kind
mm,
of another
yeah,
another example
mm,
where they did see balance.
mm. You also, I think, mentioned because I think in the 2000 paper you have two models. One where the excitatory and inhibitory neurons are the same, I think, and they have the same parameters, and one where they are somewhat different. I think you called it Model B or something. And then in that case, you there will be some phase lag between the, I guess, in the oscillatory states then, between sort of excitatory and inhibitory populations. You mentioned that as a possible test. I'm not quite sure if this has been looked for or seen or.
So phase shifts between different groups of neurons, it's been characterized in some structures, yes. For example, in the hippocampus, in
Mm,
different oscillations, people have characterized phase shifts. So then it's more complicated because inhibitory neurons are subdivided into multiple subclasses. And so the phase shifts depend on which population
mm,
of inhibitory neuron you're recording from. But I think in general, the expectation if you have an oscillation that's due to excitatory inhibitory feedback loop, in most cases you expect that inhibitory neurons will be firing after excitatory neurons, so there will be a phase lag. Um,
mm,
and I think that's broadly consistent with evidence, at least in the hippocampus.
mm.
Yes.
So, but what about like if you sort of now the functional advantages, everything of disadvantage of being in a balanced state? I think one thing is that you already mentioned is fast tracking of stimulus input if you already want to change your activity. And because I've been thinking also, if you're standing, if you are like, you want to show off your car in like in a waiting for green light in a crossing, then if you really want to accelerate, you want to have the gas pedal and the brake pedal in at the same time, and so that when the light goes from red to green, you can just release the brake pedal and you sort of jumps over the cross and impresses all the. So maybe it's sort of illustrating this the, the cleverness of that is much faster than if you have to start from with only the gas pedal, maybe. So
Yeah,
maybe
I mean,
that's a
the basic idea is that if the network was completely silent when you have a sensory stimulus that comes in, then
mm,
necessarily, so first you won't be able to detect very weak stimuli because if they are
mm,
neurons won't
yeah,
make it to threshold, so that's like a drawback, not being able
yeah,
to detect weak stimuli. The second thing is that it will take time to bring the membrane potential to threshold. Of course, if your
yeah,
stimulus is super strong, that time will be short,
yeah,
but there
mm,
will be some minimal time.
yeah,
If you are
mm,
in a balanced state, there are always at any time there is some fraction of neurons that are actually sitting close to threshold,
yeah,
and
mm,
so then first you can detect now really weak inputs,
mm,
and the second thing is that you can detect them very fast because
mm,
you're ready to cross. Some neurons
yeah,
are always
yeah,
ready to cross threshold, basically.
yeah. So
So
instead of
that's
them,
a
yeah,
big advantage.
instead of being at like the membrane time constant, which is maybe like
Right,
some tens of
exactly,
milliseconds, it's like the synaptic time constant or fast. So it's like much shorter. What about
exactly.
like things like all this to code things efficiently and compute things robustly and is that something which is obviously is it advantages to be in a balanced state from that? I'm must quite sure this must have been studied.
Right. So I think there's been like a different lines of work in this direction. So there is, I think there's beautiful work by Sophie Deneve and collaborators, where
Mm,
the idea is that if you, so what she found is that if you have like very strong and very fast inhibition that kind of cancels, constantly trying to cancel the main inputs,
mm,
then you can code much more efficiently external inputs.
mm,
And so that's a very
huh,
intriguing hypothesis. So I don't know of any evidence, I mean, showing that
mm,
this happens.
yeah,
And also it relies on the fact that inhibition should be able to track quasi instantaneously, the excitatory network activity. So I'm not sure about whether this will work with realistic time constants. Actually, this might have been done in some follow-up paper that I haven't
mm,
that I haven't
yeah,
read. But in any case, this is a very interesting hypothesis.
mm,
And then there is work by different groups showing that networks that are in a balanced state can basically easily generate selectivity to external inputs, right? And
mm,
I think the key idea here is again the idea that these networks are very sensitive to fluctuations around their mean, because the idea is that inhibition cancels the mean, and then what neurons pick up are really fluctuations around the mean. And so there are several groups that have shown, I think, the first ones were actually Carl von Rijswijk and David Hansel, and maybe and I think Helman Mato. They showed that you can get, for example, very strong orientation selectivity in a network that has no, completely random network, and weakly tuned inputs, because the two previously known scenarios, where one scenario will get orientation selectivity because you have strongly tuned inputs from the thalamus, so that's the Hubel and Wieser scenario.
mm,
But then the other scenario is that maybe you have weakly tuned input, but then you have structure, the recurrent connectivity structure is correlated with orientation tuning. So that basically the ring model scenario. And so the recurrent
mm.
connectivity amplifies selectivity from the inputs.
That's like this Ben Yishai
Right. So
idea,
Ben
yeah,
Yishai,
from 1995 or something. Yeah,
exactly.
and Sopolinsky. Mm, yeah,
And what Carl, David, Hellman, and also actually there was a similar paper by Chaim Sompolinsky and Cengiz Pelevan, where they showed is that now you can have weakly tuned inputs and a random network, but in a balanced state. And
yeah,
then what happens is that if the network was not in a balanced state, basically neurons would not be selective at all, because they will receive many uncorrelated inputs, and so
mm,
again you would have the central limit theorem and really small fluctuations around the mean. But if you're in this balanced regime, inhibition cancels the mean, the fluctuations are much bigger, and now you're sensitive to fluctuation, and you can get orientation tuning using this mechanism.
Hmm.
So that's another, and this could actually apply to many contexts where you have even maybe a weak input that's not the primary input coming into an area, but still the network will be able to encode that input because it gives some fluctuation around the mean. And so in fact it was something that we studied a few years ago with Alessandro Sanseni, which at the time was a postdoc working with me, also in collaboration with Ken Miller and John Reynolds and Mark Heisted, where we showed that actually this scenario can reproduce experimental observations in V1 of both mice and monkeys.
Yeah.
And there are probably many more examples, but I think this
So this 2000 paper is mainly about like the background states, like a spontaneous activity, and say that we are in this AI state, and then I would like to model them. I would like to understand what happens when you get some stimulus input on top of that, and then I think you describe there that like often when you take this, it's the response on top of the balance state is actually quite linear. So if you double the stimulus size, I don't know what that should be, maybe some kind of visual input or whatever spot or whatever that, and then it's quite linear. So does that mean that you can get away with describing this stimulus response on top of the like the balance state in a simpler way, or like a simple firing rate model or something like that? What's the if you don't want to, of course you could simulate the balance network and then things on top of that, but that's quite complicated. So a simple firing rate model for the extra activity on top of this balance state would be simpler. Is that is that even feasible, or think that's suitable?
Well, you're right that in the limit, when you have large number of inputs in these balance states, the prediction is that the mean firing rate of the network should basically change linearly with the mean inputs to the network. Now things are more complicated if you go to the single neuron level. So this is really a statement about the mean activity.
Okay. Yeah. Okay.
And so you do in fact get a linear, or it's actually, of course, it's only linear up to, it's essentially a threshold linear or piecewise linear transfer function. Because the
For the
rates
population
are
or for the single neuron?
no, for the population.
For the
But single
population.
neurons can still have nonlinear.
Yeah. But I mean, this linear threshold, that's what they have in these AI networks, right? So maybe it's
Yes,
like
yeah,
it's more like a model for a population on top of a balanced state.
right,
Yeah, maybe they are
yeah.
cleverer
But
than we.
single
Yeah.
neurons can still have nonlinear relationship with their inputs.
What is saying? What could have the single
Well,
neurons?
the single neurons
Yeah.
still have nonlinear
Yeah.
relationships with their
Hmm.
inputs. Yeah.
But I guess if you think in terms of, well, if you believe that the population, somehow a population is sort of like the fundamental computational unit in cortex or a group of neurons, then maybe, yeah. Anyway, maybe it's sort of you can sort of model that like rather simple maybe and get away with it.
Yes, I mean the question is whether depends at which level you think, at which scale you think computations are performed,
Yeah,
of course.
obviously.
But yeah, if you're thinking about columns and having similar neurons
Yeah.
with a columnar organization with no sharing
Yeah.
similar properties,
Yeah.
then yes.
Cool.
Yeah.
We are all for simplification, and we all like linearities. At least I do. So, but then, so what are sort of the? I mean, if you go to, I think, I mean, a 2000 paper, then you had, well, this homogeneous neurons, or maybe you distinguished between excitatory and inhibitory in this model B. But I mean, so now if you sort of have you has it been studies on like now you have several inhibitory neuron types which are sort of whatever the serotonin and VIP and what it's called, these three types. So is that has there been work to sort of to extend sort of there are several ways to extend the work that you did in 2000 in terms of heterogeneous neurons and more
Yeah.
heterogeneous connections and synaptic plasticity and so on. Is that do you have an overview of what's going on?
Yeah, well, I mean, in terms of increasing the number of populations, I mean, surprisingly, to my knowledge, nobody has really, at least from the analytically, not in terms of simulations, but
Huh?
nobody has really characterized networks with this distinction between different types of inhibitory population. There are an increasing number of papers, but people typically use rate models to describe
Yeah.
these different inhibitory populations.
Yeah.
I mean, in principle, should be relatively straightforward because you would have balance equations for
Yeah.
each population. So the idea is each population will be balancing their excitatory
Yeah.
and inhibitory inputs. It could be actually that some populations may be out of balance. It could be that some populations are not in this sub-threshold poisson faring regime, but are actually in another
Hmm.
regime. So that's not to be excluded.
What
But
about using this population density approach? I mean, I think when people look at the population density approach, and if they don't have a background from like statistical physics or physics, then it looks a bit daunting. It's a little more complicated. So doing this bifurcation analysis is maybe more of a like conceptually and technically a little bit more challenging. But there's maybe there should be possible to make some like automatic tools for doing this or something
yes,
to so that we
yeah,
don't have to do it by hand.
yes, definitely. So, and this is something I've been thinking about for some time. I just need to find the right people to do it.
Yeah.
But yes, I think
Okay,
it would be
maybe
very
maybe the
helpful.
right person is listening to you. So contact contact Nicolas.
No,
If
I agree because these are quite, I mean, complicated to implement
yeah,
if you're not familiar
yeah,
with the theory. And
yeah,
so, yeah, I agree. It would be nice to have a tool where you specify you have these different populations and you
uh,
enter all
yeah,
parameters, and it tells you, for
mm,
these parameters, you will be in an asynchronous state, or maybe
uh,
you are in an oscillatory state, or this kind
yeah,
of tool. I think would be very
yeah.
nice.
Because there
But
are a
and
few.
it's certainly doable. So there are two actually. There is one difficulty here, and the difficulty is that so one has to keep in mind that these calculations are always an approximation because you're doing basically this diffusion approximation,
Mm,
um, and you're assuming that neurons are independent,
mm,
or they can be only correlated through the global firing rate of the different populations in the network. That's the only source of correlation in the
mm,
in these calculations. The other thing is that really these calculations can be performed exactly only when you ignore synaptic time constants,
mm,
right? And with the simplest model, with the leaky integrated fire model,
mm,
as soon as you add more currents to your model, let's say you want to add an adaptation current or
mm,
you want to implement realistic synaptic time constants, then everything becomes much more complicated. There
mm,
are various approximations that are present in the literature, but it's so again it would be actually be useful to have like one place where people can see what
mm.
everything has been done and
Yeah.
try various
Because
approximations.
I think Mark de Camps in Leeds, he has been making this population density. Uh, uh, what I think it was called. I don't remember what it's called, but it's at least there you're able to go beyond this diffusion approximation, so you can sort of make sort of like finite larger steps. So it's maybe more suitable if you are in a brain region where you actually get some inputs which are much stronger.
Are you
But
talking about?
I
I missed the name. Magnus Richardson. Are you
no
talking about?
Mark the Camps
Oh, okay, okay.
Leeds.
No,
Yeah, Mark
because
the Camps. Yeah,
because Magnus Richardson has also
yeah,
worked on this on the issue. What happens if you have instead of
larger. Yeah.
small PSPs if you have like
Mm.
finite PSPs. Yeah.
Because I mean, even if you're not, you could be able to identify these bifurcations numerically, I guess, also, right? I mean, it's that you don't. So
Yes.
if you have like, it seems to be like a, if you could get like a general tool that could do this, it would be useful for the field.
Yeah, no,
Mm.
for sure. And you can, you could increase. So basically, these systems are described by essentially partial differential equations. So when you have finite jumps, it's more complicated because it's not a PDE technically; it's a differential equation.
Mm.
So that's which is
Yeah,
more complicated. But you could imagine having tools to simulate numerically
yeah, yeah,
these
yeah,
equations.
yeah. Exactly that. I think the tool of Mark de Camps is called Mind, MIIND. Yeah,
Mhm.
so that's
Okay.
anyway. So yeah, we have sort of a. I'm a sort of a friend of his. So I know a little bit of his work. So also, I mean, well, how far do you? I mean, I think when you sort of, I mean, so far we talked about like cortex, maybe like sensory cortex is where you have most. Well, I guess also cortex and hippocampus. But I mean, if you go out towards the periphery, for example, in the thalamus, do you think this balanced ID works in this sort of more more nuclei, maybe more closer to the to the sensory outside, and if they are how where do you expect to see balanced networks, and where do you expect to see something else?
Yeah, good question. I think more generally, I would expect this kind of regime to happen in networks where you have um, neurons with lots of inputs and lots of weak inputs. And of course, inhibition is a key ingredient, but I mean, inhibition is of course ubiquitous.
Yeah,
Um, but there are neurons that don't really fire close to Poisson processes in many brain structures. I mean, Purkinje
yeah,
cells in the cerebellum. I mean,
yeah,
several types of cells in the basal ganglia. They
yeah,
typically neurons that fire at pretty high rates. They
yeah, and
are
maybe
brain
they're
structures
fired
that,
by
of
them.
course, generate very strong oscillations,
Yeah,
like networks that control various physiological rhythms. So there, of
yeah,
course, it's not clearly not a balanced network.
yeah.
Pretty
So I
far from
guess
it.
exactly neurons that fire by themselves, so without an input, like
Mm.
some of this.
Yeah.
Yeah. So what about like any pathologies associated with the breaking of balance? Maybe epilepsy is sort of epileptic seizures. Could that be a break of balance disease or phenomenon?
Yeah, I think it's really an obvious candidate uh, for what goes wrong in epilepsy is basically that your inhibition is no longer strong enough to stabilize the network, or you can have these kind of brief instabilities that show up. Um, I frankly I don't know about other
No.
um, whether it could be linked to other pathologies. There are
Yeah.
certainly other pathologies where you get oscillatory instabilities like Parkinson's disease, but I don't think
Yeah.
it's clear
Yeah.
really what happens, even what is mechanistically the what kind of synaptic links are
Yeah.
involved in
Yes,
these
an
oscillations.
alternative thing is that it could be like there's some kind of circuit connections between different nuclei and stuff that maybe sets up gets faulty, so that you get these oscillations. Yeah. So,
Yeah.
so I mean, this just now a little bit sort of towards the end here, but I mean, these papers, at least the ones I've talked about, they came. I mean, this 2000 paper came out 25 years ago, so it's sort of a yeah, it's you're getting
Already,
old. We
already
are
interesting.
getting old, but it's so. But I mean, of course, you have worked on this also more recently, I guess. But you have also worked on other things in the 25 years that has passed, right? So what has been your what is your main interest now, for example, or do you have several interests? Or
Yeah, so I think my interest. So I've been my main interests have always been more related to learning and memory. But as I was trying to investigate more and more realistic models, there were kind of these side questions that that kind of distracted me for
Yeah.
for a very long time. In particular, when I started to look at networks of spiking neurons, seeing all these kind of phenomena, and just trying to understand them. And of course, that has nothing to do with learning and memory.
Yeah.
But occasionally, I try to come back, and so and
Yeah.
yeah, I've been so there's several directions I can mention several directions. So once I had a little bit clarified what happens in networks of spiking neurons. I mean, I think we still barely scratched the surface. There's still lots of things to understand, but the next step would be to study what happens when you include synaptic plasticity. And a number of people have actually worked on this. When I was about to start this, actually about 20 years ago now, or maybe 15, 20 years ago, but then I realized I was not really. The question is what kind of synaptic plasticity rule you will insert in these networks, right?
Yeah.
That's the first question. And then at the time it was not really clear what kind of plasticity rule to include, because at that time by far the most popular rules in networks of spiking neurons were the spike timing dependent plasticity rules. That were There were like tons of papers coming out with STDP rules. But the standard STDP rules turn out to not describe experimental data very well when you look carefully at the literature. I mean, they describe very specific experiments where neurons fire basically periodically, both pre and post, with a specific delta t. Of course, that's totally not physiological. Neurons don't do that at all in cortex or hippocampus. Then, if you look at other experiments where people do different patterns, activated using different patterns, you find out that these rules don't describe really very well plasticity. So I've been working on basically models of synaptic plasticity, and in particular, one direction was to try to derive a kind of minimal biophysical model for plasticity that I think a little bit in the spirit of the leaky integrated fire neuron. So in the leaky integrated fire neuron, you try to include like what's the key variable, which is the membrane potential, and then you add a threshold, and that's it. So that's very minimal. And so for synaptic plasticity, one key variable is postsynaptic calcium concentration, right? Because if you block calcium influx into the postsynaptic side, you typically block plasticity in most experiments. And so basically trying to build simplified models of this type, where you have basically a variable describing your calcium concentration on the postsynaptic side. And so it's influenced by both pre and postsynaptic spikes, right? Because the presynaptic spike, because you have glutamate binding onto NMDA receptors and NMDA receptors opening and calcium influx. Postsynaptic side, because it can affect NMDA receptors through the voltage dependence, and also it opens calcium channels. Basically, if you assume there is a backpropagating action potential. And then you can add thresholds. It's really in the same spirit as the leaky integrated fire model. Basically, you add a threshold for plasticity, and there, except that you have two thresholds: one for depression and one for potentiation. And actually, we found that this model can really go a long way into capturing lots of different experimental data. The nice thing about it is that it automatically captures some non-trivial features of the data. For instance, so it can capture all kinds of so-called spike timing dependent plasticity rules that describe how plasticity depends on the time difference between pre and post. But once you are in a parameter region where it does produce a particular type of plasticity rule, then if you use other patterns, then it kind of automatically reproduces the phenomenology. Like if you use triplets instead of pairs of spikes, for instance, then it kind of gets at least qualitatively the right behavior. And same for the firing rate dependence, it's kind of pretty much automatically.
So these are then rules that like STDP rules that can sort of people can use then, and maybe that you
Right.
use.
Yeah. So the idea is that then what one could try to insert these rules in large networks. So of course now the problem is that the drawback is that these rules are more complex than just standard STDP rules, where some people have made progress on trying to understand analytically how implementing a STDP rule in a large network gives rise to various types of network structures.
But you'd like to stay in the balanced state, right? So then, while doing this, but I guess this at least one paper that I really liked was the paper from Gerstner's group with the Vogels and Spreckler and Senke
Yes.
maybe, about where they had like this inhibitor plus plasticity, which self-tune the network to get to the
Yeah.
balanced state. So that's maybe also nice to put in there then.
Yes, so that's always that's where it gets complicated. Is that typically when you're using only a Hebbian rule for excitatory to excitatory plasticity, you often get all kinds of instabilities, and then you have to add some stabilization mechanism. And this is, I mean, this inhibitory to excitatory plasticity rule is one potential
Mm.
stabilization mechanism.
Yeah, but you
And
don't,
it,
you
and
haven't
it
talked
has
about.
other nice properties as they showed in that paper. So
Yeah. So, but you don't talk. I mean, short term, like pair pulse depression and what a pair spike depression was called pair pulse. So these things are not. Are they? What about those? I mean, they are. Are they? Could they be included? They haven't been explored much, or
no, they have been explored. Um, yeah, by actually lots of people. So I mean, there is of course the work of Misha Tsodix and collaborators on population spikes.
okay,
Um,
in a
you can,
balanced network setting, sort of.
yeah,
Mm.
pretty much.
Yeah.
And also potential roles in maintaining information in short-term memory. The idea is that
Okay.
instead of having persistent activity,
Uh,
keeping the information in memory, you have basically synaptic variables, something like
mm.
calcium
Yeah.
accumulation in the presynaptic
Mm.
terminal, and
Yeah.
and so you can get another form of another mechanism for short-term memory, which relies on synaptic variables instead of
Yeah,
neuronal
we had
variables.
actually for regular listeners, we just had a podcast with we had a podcast with Pavel Herman. Earlier
Oh,
last year maybe about this or like this kind
okay,
of approach, new approach for
okay.
understanding short-term memory with the synaptic variables. So, I mean, one thing you have not used so much, maybe, or what do I know? Well, haven't looked so much at biophysically detailed neurons or neurons with complicated dendrites, or maybe you have. So, so the obvious reason for dealing with point neurons, like integrated fire neurons, but what role do you think what are the key things that you miss by looking at point neurons? You think?
Well, I mean, for sure, you miss a lot of potential interesting computations that happens when dendrites are nonlinear, have nonlinearities, and we know that potentially they can have nonlinearities. Um, and um, and that's very much something I have in the back of my mind, but there's only a,
It's, it's
a
something you
finite,
worry about, but
finite amount of things that you
yeah, no, it's
that you can work
absolutely.
on.
Yeah, yeah.
And
No,
no, I mean
that's true.
something that I have to mention actually, which is I think we should definitely mention this in the podcast on balanced networks, is the fact that typical balanced network models consider so-called current-based synaptic inputs, where in the large k limit, so again the limit when you have a large number of inputs, the synaptic inputs to both excitatory and inhibitory inputs kind of grow unbounded because there is no
Mm.
reversal potential implemented in this. And of course, it's been from the very beginning, people have worried that, well, in reality, synaptic inputs cannot go to plus or and minus
No,
infinity.
exactly.
That's clearly not
No.
possible. And
Mm.
so, what happens then when you use conductance-based
Yeah,
synaptic
yeah.
conductance-based description for synaptic currents? And things are kind of start to become a little bit different. So that's something work that actually again Alessandro Sonseni, who was working as a postdoc at Duke, he's worked on the different types of scaling laws that you need to implement between
Wow.
the amplitude of postsynaptic potentials and the number of inputs for to get a balanced state. And you do get a qualitatively different regime
Wow.
in networks with conductance-based synaptic inputs.
But the still point neurons, or
They are still point neurons, but
mm,
then the question is, now if you have, so that the next question about, is like synaptic inputs, you have basically its conductance changes, but now they are scattered all around a spatially
uh,
extended analytic tree. And I think there are really interesting questions there about
yeah, that's true. It's
what
not quite.
happens.
So I guess making a conductance-based neuron synapses on point neurons is not clear, obvious to me at least, that this is some sense more closer to truth than since you than
Right.
current based neurons, since you don't take this dendritic tree into account.
Right, exactly. So, and in
It's
fact, that
has to
brings
be
it
checked.
exactly. And so that the question is, what happens if you have like
Hmm.
conductance changes scattered around the dendrite, and
Yeah,
then
that sort of could be a bit
it's not
different situation.
it's not a current-based point neuron. It's not a conductance-based point neuron.
Yeah.
It's something different.
So, but I mean, so I think when we started off with this, when you say that, like back in the days when most physicists, or most, yeah, I guess they were most physicists anyway, who worked on this, like Hopfield and statistical mechanics, in building on the work of Hopfield, for example. And then they didn't really read too many experiments. They stayed in their little sort of like community, which is understandable. And you said you didn't read an experimental paper before you sort of got to Rome with Amit or something. And of course, I remember when I started doing neuroscience, and it's quite overwhelming to start to looking into the experimental literature and try to identify a problem.
Yeah.
So, and how to keep track of the whatever, I mean, keep track of the literature, and so how this, now you're getting so old that you have to come with advice to young researchers. And for example, how do you how do you choose a problem if you are a young scientist, and you want to work close with experiments, maybe electrophysiology or like the neuroscience experiments? So how do you what's your recommendation? What are your thoughts, tips to the next generation?
Well, that's a tough question.
Yeah.
Well, I guess what I would recommend is that if you're like a theorist, to first to talk to the nearest experimentalists, right? That's kind of invaluable to have, to be in a place where you have both theorists and experimentalists. And when you see the phenomenon that's unexplained by current theories and something surprising, and that would be a good starting point, and then start to understand what kind of models could potentially describe this phenomenon. And
Yeah.
I think there is no, and this can be applied to experimental phenomena at very different levels, right?
Yeah.
From the really
Yes.
cellular level to the
Mm.
to networks, or even at even larger scales.
Yes, I guess one good thing now compared to when you started is that now there's a lot of experimental data available online.
Right.
Of course, that is, you have to learn what they mean, and it's like it's not that
Right.
easy to use them. But nevertheless, you don't have to be in an experimental lab or next to an experimental lab in order to have data to work on.
Yeah.
Of course, it still helps to talk to the experimentalists who did the experiments, and sort of to be in a, well, talk to
Mhm.
experimentalists. But at least that's one, the
Yes.
technical, that issue is not so
Yes,
certain to see.
that's a huge improvement. And I remember back in the days, it was very hard to get experimental data if you were not a collaborator of,
Yeah. Yeah.
and often before, of course,
Mm.
before publication. Sometimes even after publication, people would not give
Mm.
access to their data.
Yeah.
Also, I think something else, I think, which is a big improvement, is now you have all these online. There is this, I think it's still ongoing, right? The Neuromatch Academy. So
Yeah,
you can actually
that's true.
learn.
Yeah,
about these things and learn about data analysis methods and also, of course,
yeah,
computational
that is
models.
true. Hmm.
And so
So
that's also
as long as you have
important.
an internet connection and is so you can sit anywhere in the world, just like listening to this podcast. I hope it's really anywhere in the world, not just that big research institution or labs, maybe in the Europe or North America or like
Yeah.
certain parts of the world. So that is we have a lot of brains that can be mobilized. So yeah, so that's a good thing. So yeah, I don't know if there's something you feel that we haven't talked about.
Um, no, I think we've covered a lot of ground.
Yeah, that's good.
Probably,
I think so too. But what about
I'm
AI?
sure I'm sure we missed lots of important things, but I mean, at
That's
least we
true.
covered.
But I mean, this is of course there will be links to papers, at least the papers we have talked about on the homepage of the episode. So
Okay,
that's yeah. So that's.
so
But what
very
about
nice.
like, do you use AI in your research? Well, how do you say AI changing your yeah?
Yeah, so I don't use AI personally. I'm actually in a department where lots of people are using AI, and actually
Mm.
lots of people are trying to understand how AI works, which is a big
Yeah,
a big open
yeah,
question nowadays.
yeah,
But sure, I mean there are many AI is, I think, revolutionizing neuroscience. I mean, especially data analysis in neuroscience, right? There are
yeah,
lots of things that you can do that you could not do um, basically just 10 years ago. Like one kind of first example that come to mind is the connectomics, where
yeah,
without automatic segmentation tools and
yeah,
then you will not be able to reconstruct the kind of volumes that people can reconstruct nowadays, which
mm,
apparently you still need to do some proofreading, unfortunately. But
yeah, mm,
um,
yeah,
and I'm sure we're just at the beginning of, like, I think
yeah,
AI will of course revolutionize
yeah.
neuroscience. That's for sure.
Mm. Actually, before
But
this, when I planned this podcast with you, of course, I had thought about it for some time what I want to talk about. But then, just for the fun of it, this weekend, I asked my large language model, and that I'm going to have podcasts with you about this and the balanced networks, and maybe starting with your work with Daniel Amit. And actually, the suggestion it came up with was pretty good, actually. Not very difficult from what I would have done anyway. So it's almost a bit scary because it comes after 1 second, right? It's almost a bit spooky. I think how fast it comes, how efficient they are, and how good they are at these things, right? Of course, you have to look through it with a bit of, you have to check it, but
right,
nevertheless, it's
yeah,
pretty impressive, right? So
no,
yeah,
it's always important to check because
that's true.
sometimes these
So
systems can fail spectacularly. So
absolutely. So anyway, so thanks a lot for coming to the podcast, Nicola.
very welcome, Gote. Hope that next time we can meet in person some meeting.
Absolutely, and then you'll get a cup. Actually, every all I think
Oh,
I all guests get this theoretical neuroscience cup. Of course, it's a bit difficult to bring them all, but so next time I'll bring a cup. I also have bunch of other guests who I owe a cup. So that's I have to travel around the world now with cups to make up for myself. And yeah, absolutely. Okay, thanks. Bye.
right. Bye.